Hello and welcome, everyone. I am Christy Zono, Director of Genetics and Health Policy at Genetic Alliance. Today, as part of our Hot Topics in Genetics and Advocacy, our presenters will be talking about the Therapeutics for Rare and Neglected Diseases, or TRND, trend, a new NIH program. Our presenters today will be Steve Groft and Melissa Ashlock. Our first presenter today will be Steve Groft. Steve is the Director of the Office of Rare Diseases Research at the National Institutes of Health, or NIH. His major focus is on stimulating research with rare diseases and developing information about rare diseases and conditions. To help identify research opportunities and establish research priorities, the office has co-sponsored over 800 rare disease-related scientific conferences with the NIH research institutes and centers. Thank you, Steve, for joining us today.
This whole concept and need was initiated during the period of June and July of 2008 when Dr. Zerhouni discussed the possible new NIH initiative to develop therapies for rare and neglected diseases with members of Congress. This has been a major concern for a number of years, as people in the audience there who are listening will attest to. And so in July of 2008, there was language inserted into the House and Senate fiscal year 2009 budget for the NIH that funds would be made available to jumpstart a rare and neglected diseases initiative. And after that, we sort of waited for the budget to be passed, but it was not forthcoming immediately, but the language was still there. And so during the period of October and November of 2008, we started to prepare a 5-year budget that would at least give us an opportunity to initiate the program and gain some experience, and then during the 4th and 5th year, towards the end of the 4th year and into the 5th year, would enable us to do an evaluation of the progress and some of the strong points or the weak points that we may have confronted as we were implementing the program. And so finally, in March of 2009, the budget was approved, and approximately $24 million was provided for this activity for fiscal year 2009. And we anticipate that this amount of money will continue to be made available on a yearly basis. So it's just not a one-time $24 million set aside that would be used for a 5-year period, but rather would be approximately $24 million per year for the next 5 years. And these projects will not be some of your typical 6 months or 3 months type studies. They will be individual projects that could take up to 3 years to conduct, because they will involve various types of studies, and I think Melissa will talk about these in a little bit more detail. They will be monitored closely for progress, the same as what the pharmaceutical industry would do, and those that do not make sufficient headway, we will terminate them, or if they're showing that there's any signs of toxicity or other decision points that we would say, the animal studies indicate that there are some potential problems, we'll look at those very closely and then try to make a decision, a go or no-go decision, how much further we keep working towards this. As came out in the press release, this is a joint program between the NIH and all of the institutes and the Office of Rare Diseases Research. The program itself will be centered within the National Human Genome Research Institute, and funds that will be made available will come from all of the institutes and be made available for these individual projects within the program. And we will have several different advisory groups or review groups, if you will, that will provide the oversight and the governance to the whole program. And so we will have 2 separate groups. One is a Trans NIH Staff Advisory Group with members nominated by the institutes and centers here at NIH. That will sort of be our liaison back to the institutes, and they will also keep us informed of how we're going to work with individual institutes and look at other programs that either will feed into what we're doing from, say, for example, the Molecular Libraries Initiative, or that will flow out of this into clinical research studies. And so we'll be using that group to help us look at the activities and the plans that we're putting together. And we also will have an external expert external panel, and that will be comprised of individuals from primarily outside of the government who have a focus on product development and research. Certainly this is an area that some parts of NIH has considerable experience. Some of the groups such as the Cancer Institute, the National Institute of Allergy and Infectious Diseases, and National Institute of Neurological Disorders and Stroke, several of these groups, institutes already have product development programs. And so, but we want to get the expertise of individuals, some from industry, who've been involved in preclinical development of products, because it's a whole different world in the preclinical development, looking at the results from animal studies, looking at the development of compounds, utilizing the services of medicinal chemists to try to determine the best possible product that should be going into humans. And so we are in the planning stages now. We've solicited nominations from the institutes, and we are at the point now of making selections of individuals to serve in these capacities with these advisory groups to the program. And so if we can have the slides, I'll turn it over to Melissa and let her take it from here.
Okay, thank you, Steve, very much.
Great. I'm just going to introduce Dr. Melissa Ashlock, who is Vice President of Drug Discovery for the Cystic Fibrosis Foundation, Therapeutics, the Drug Discovery and Development affiliate of the Cystic Fibrosis Foundation. And her roles include overseeing projects directed towards discovering and developing drugs that treat cystic fibrosis and using genetics and genomics-based approaches to discover novel cystic fibrosis drug targets. Prior to this, she worked for 5 years as an investigator at the National Human Genome Research Institute at NIH, where she was acting chief of the Vector Development Section. And prior to that, she was a scientist and medical staff fellow in the pulmonary branch of the National Heart, Lung, and Blood Institute of NIH. She received her MD from Cornell University Medical College and completed her internship at New York Hospital. She's board certified in internal medicine and did her residency for that at Mary Hitchcock Hospital at Dartmouth College.
Thank you. So I work with the Cystic Fibrosis Foundation for several years, and I'm in the process of transitioning, working with the NIH. So I'm going to talk to you a little bit about the initiatives related to rare disease drug discovery and development from 2 points of view. One, from the perspective that I have from the Cystic Fibrosis Foundation experience as director and then vice president of drug discovery, and the other in my role helping set up this new Therapeutics for Rare and Neglected Disease initiative that Steve described. And I'm really reporting to you on behalf of not only Steve, but also Chris Austin. And what we had planned to do was to talk to you about, first about what really the need is, and then to describe the trend initiative to you. So my first slide, basically, the problem that we have in drug discovery and development is that for rare and neglected diseases and for diseases in general, there's a difficulty translating basic research findings into clinical candidates that can be tested in the clinic in individuals with rare diseases. And that's really shown on our first slide with a picture of the San Francisco Bay. The idea is that we're trying to build a bridge between the basic research issues and the clinical trials. And so we don't want to focus too much on this. It's just a way of showing you that there can be a bridge that's built, and what we're trying to do is do that. On the second slide, there's a picture here of the human genome nature issue, which basically helps us understand the various targets that could be contained within the human genome. And over the course of several years now, we're getting much more information about what the potential genetic mutations are that cause disease, and the proteins that are produced by those mutations could be biologic targets for drugs. what this is basically showing us is that not much of what is available has been exploited. And so one of the goals that we have at NIH and many people do around the world is to try to exploit this new information. On the next slide, the problem is really described here with respect to how little has been done and what the opportunities are. And the title is just describing that a small percentage of diseases and a small percentage of genome-encoded targets are being addressed. And on the left, if you think about the genome as the whole pie, that small blue slice are the current drug targets that are pretty well understood for which drugs have been developed or are in development. And on the right-hand side is a pie for human diseases. And you can see in the little yellow slice, that's the diseases for which pharmaceuticals are available or in development. And those are predominantly for prevalent diseases that affect the developed world. And the rest of the pie are diseases that affect millions and millions of people for which no work has been done, or if the work has been done, it hasn't been translated effectively into drugs. On the next slide, the problem is described in more detail. So there are 7,000 diseases that are known to affect humankind, probably more than that. A small fraction are commercially attractive to pharma. And when we think of neglected diseases, there are really two types. One is those that are low prevalence, the rare diseases of which you all are familiar, and there are probably about 6,000 of those, probably more. And about 25 million people are affected by those. And often the etiology can be traced to mutations in the human genome. And common examples of this are cystic fibrosis, SMA, sickle cell anemia. We could go on and on about that. The high prevalence diseases that are neglected are those in the developing world. And they may be rare in the U.S. because usually these diseases might be acquired by people from the U.S. or other developed countries when they travel to these undeveloped countries. So they're high prevalence in the developing world. They might be rare in the U.S. Whatever the issue is in the developing world, they cannot pay for the medicines. And most of these are infectious diseases. And so even though their targets may be known or there may be drugs that could be developed that could be effective, they're not commercially viable. So companies aren't really spending much time or money. On this next slide, the current drug development paradigm is a slide that basically shows in cartoon form how you could think about drug development. And it starts with the science on the left and goes to the clinical testing on the right. And we'll just walk through this. The first idea is that you need to figure out, if you're going to discover a drug, what it is the drug is going to target. And that doesn't actually mean the drug has to bind to that target. It really just means that the drug has to cause something to change in that aspect of the disease. So I can use cystic fibrosis as an example. What we targeted with cystic fibrosis is the protein function, which is chloride transport. So you then need to develop an assay that could be used to identify compounds that affect that target. So in the CF case, the assay had to do with whether chloride was moved from inside to outside of a cell. And then you use information about the target and assays that you devise to identify compounds. And the first set of compounds are usually what we would call tool compounds. And that's really the purview of the NCGC, and I'll talk a little more about that in a minute. But you could use this information to develop assays to find tool compounds and do that either knowing the structures of the proteins, and that's the ribbon-like diagram there, or using a combination of that information plus screening with robotic technology, and that's what that robot is shown right below the ribbon-like diagram. The next thing is to optimize the compounds that are identified and do this for activity against whatever target you care about. And in the case of cystic fibrosis, the optimization has been done using medicinal chemistry, which is a branch of chemistry that is a synthetic approach to make compounds look like drugs by doing chemical synthesis and modifying different parts of the compound. And when this is done, you try to maintain activity and not and also improve the pharmaceutical properties. And there's a Rubik's Cube shown here because this is often a process where you change one thing to increase the pharmaceutical profile, but you lose activity. So it's a real art for the medicinal chemist to try to take a tool compound and make it into an efficacious, safe compound for testing in animals. And then finally, when we have a compound that's been tested and looks safe and efficacious, and an investigational new drug application has been made and approved by the FDA, clinical testing can be done in humans. So one of the obstacles to this, besides the scientific issue, is to be able to recruit patients to be studied for these trials, and also to make sure that what is being studied in these clinical trials as an endpoint is relevant to the disease and also relevant to the way the compounds were discovered. So, for example, in cystic fibrosis, the discovery process involves the movement of chloride, and in the clinic, the outcome measures that assess the activity involve the movement of chloride. So there was a one-to-one relationship of what was used as an assay in the lab to what was used as a measure of how efficacious the compounds were in the clinic. So on the next slide, I just wanted to give you a quick primer with clinical trials, and probably most people know this, but just to give you an idea that clinical trials are a stepwise process that seem easy to describe, but are the most resource intensive of the entire process. And that's because they require clinical grade material for which manufacturing and quality assurance is very expensive often, and then they involve initiating clinical trials often around the world where you have to pay to perform the trials, but not only perform the trials, get everything set up for the efficacious implementation and data collection. And so on this slide, you can see phase 1 in red. These are the short, small trials for safety and dosing. It's usually the first time a therapy is tested in humans, and it's usually tested in healthy individuals. In this case, I had cystic fibrosis, but whatever disease you're interested in. In yellow, I put this kind of as caution because if you move from phase 1 to phase 2, you're doing pretty well. These are medium-sized trials that are of medium duration. Just as an example, in CF, the size has often been about 40 patients and the duration 4 weeks. And the concept is to look for safety and find proof of concept. Does the drug candidate modulate the target? So does the chloride move out of the cell? Or does it move out of the airway? And then once these phase 2 trials are successful, then there's an opportunity often to move to a pivotal or registration trial. These are the largest, longest trials and that generate the primary data for an FDA decision. And their decision is based on, is the drug candidate safe and clinically efficacious? And these phases are well worked out and well-trodden territory for diseases that are very common and for which there are many patients. But the process is more difficult to perform when there are fewer patients and specifically fewer patients who qualify for particular diseases. So one area we're working on is trying to understand how to work with the regulatory agencies to help people understand how to effectively design clinical trials. So this is a diversion about clinical trials, but I wanted just to make sure people understood that it doesn't help too much to do this drug discovery if you don't have this aspect set up. Okay, so the next slide, I'm going to flip through here to show the conventional roles of the public and private sector on biopharm and drug development. This will set the stage of why we have started Trend and how it fits together with what's going on already. So on this slide, on the left-hand side, you can see the private or NIH-funded basic science. And many disease foundations and the Genetic Alliance and other groups have funded basic science research and have developed ways of making assays through target identification. And NIH funds this research as well. And then in the kind of pink area, you can see that in general, pharma and biotech have covered this next stage, which is quite long, several years long, and culminates in clinical trials. And then at that point, the FDA would potentially approve a drug. So really, biotech and pharma for most diseases really covers this large spectrum and large resource-intensive time. So before the Molecular Libraries Initiative, the next slide basically shows some concepts about probability of success and cumulative cost. And we'll ignore for a minute the before the Molecular Libraries Initiative because that isn't relevant to what I'm about to say. But what I wanted to say here was that the cost in the middle panel that you can see in the yellow line reflects what I said earlier. The cost of this drug discovery isn't so high until you actually get into the phase 1 clinical trials. And on the top, the probability of success is very, very different. So at the early stages of the drug discovery process, the probability of success is not particularly high. This is kind of written in a backwards way I'm seeing here. But the probability of success is low when the cost is low, and the probability of success is a lot higher when the cost gets higher. But nevertheless, the pharmaceutical industry thinks that it's an opportunity lost if they were to invest when the probability of success is low, even though the cost is low. So what we want to try to do is to de-risk things at these early stages, and that's what groups like the CF Foundation have done and other foundations, but the federal government could also do that as well. So I'm going to talk a little more about that. So the Molecular Libraries Initiative has expanded the role of the public sector from the early target identification all the way through to screening, and this is where the NCGC and the Molecular Libraries Initiative around the country has helped, so that besides just identifying targets, we now, from the public sector, have assays and screening and compounds that have been identified that are tool compounds. I wrote here venture philanthropy as a way for the private sector from disease-focused foundations to de-risk this process at early stages. So money put in at these early stages where the success likelihood is low, but the cost is also not that high, will enable people in these areas of rare and neglected diseases to get some progress so that they could hand off these compounds at later stages to pharmaceutical companies. So just go on here to say, how can the NIH help? The NIH can help to increase drug discovery and development at this early stage for rare and neglected diseases where the opportunity costs are high for industry. And we can do that through the NCGC, which is very active in tool compound discovery, and through this new initiative called TREND, which Steve has introduced, and I'll describe a little bit more. And NIH can help by discovering not only novel drug candidates, but by exploring repurposed drugs that have been approved for existing diseases, for existing drugs that have been approved for diseases already. So I'll go through a couple of examples of that. So what does the NCGC do already to help? The NCGC is a center in Rockville that was founded as part of the roadmap, and Chris Austin is the director of this, and he's also been very instrumental in getting the connection between this and the next step, which is TREND. He oversees about 65 scientists, and they've performed about 100 collaborations with investigators around the world. And those investigators have generated projects that are funded in various ways or that are originated in various ways, some from NIH, some from foundations, research consortia, and some from pharmaceutical and biotech companies, and some from intramural NIH. And there has been a focus in this group on novel targets and those for rare and neglected diseases. And on the right, you can see the building and the big robot called the Calypsis, which is the workhorse of this place. So how do we set the stage at NIH for drug discovery and development for rare and neglected disease? Well, the NCGC produces in vitro tool compounds, and this means in vitro in this case means that they're studied in cells, not in necessarily studied or identified in animals. And in the process of doing this with this high technology, they've developed new paradigms for assay development, for screening, for informatics, and for medicinal chemistry. And also, they've taken advantage of their robotic technology and the availability of approved drugs in the form of libraries from around the world, not only the FDA, to screen approved drugs, not just chemicals. So I'm not sure I've explained this properly, but you can discover drugs in a couple of ways. One is to take libraries of chemicals that have never been shown to be drugs and find tool compounds and then improve upon their characteristics and make a novel compound that would have a new patent based on its composition and its use. Another way to develop drugs is to also take drugs that have been approved for other diseases and to test and see if they might have an impact on a known disease for which there's no therapy. And this repurposing process, which is what I just referred to, has become more interesting and appealing to people, especially as they've considered some of the drugs that are actually out there in the world have target effects, but they also have off-target effects that could be beneficial. And there recently, this week, was an article in Science by Mark Boguski about using different ways of repurposing, using information from individuals who are taking drugs to try to find ways to potentially repurpose them by reporting in registries or on the internet the kinds of effects that people have when they take a known drug. For example, if they take a known drug for a headache but their arthritis gets better, that kind of information could help drug companies and help us decide whether it would be relevant to test that particular drug for arthritis. So repurposing is important. And then the second bullet is the NCGC that I just described, setting the stage using discovery and repurposing it. But I wanted to point out, I'll use two examples, the discovery of novel candidates with the schistosomiasis on the next slide, and the repurposing in using NPC and chordoma. I don't want to go through these, but you can see that right now among the hundreds of collaborations there are a number of rare diseases and neglected diseases that Chris's group has explored to try to find tool compounds for, and make it clear here that they haven't been trying to discover drugs. They've just been working to discover tool compounds for these diseases. However, in the process of doing that, they've had some great successes for which there might be a reason to move forward for a more in-depth drug development project. So I'll talk a little bit about schistosomiasis. Again, this is an example of using the NCGC to find a novel, promising tool compound that could then be made perhaps into a drug. So schistosomiasis is a parasitic disease. It affects 250 million people, mostly in Africa. Chris and his group collaborate with a fellow named Dr. David Williams at Illinois State University who identified a new target and worked together with the NCGC to identify compounds as starting points. And this was a very interesting and exciting and high-profile project that was published in Nature Medicine, showing that this group of oxidizole compounds could be new drug leads to control schistosomiasis. So really, the details of this aren't relevant except to say you can see how clearly different the liver of the mice are with this disease. On the left, the control livers are packed full of these schistose, and in the presence of this drug, the livers look very clean and undiseased. And so this is a dramatic example of the results of screening, identifying a novel compound that when given to animals actually reverse these symptoms. So this is very promising, but from this point to become a drug to be taken by patients, there's a lot of steps to take. And so we need a mechanism for which that could happen. And that's potentially the kind of thing that Trend could do. So then on the next slide, I'll just use the approved drug repositories to explain what could be done for approved drugs. So the schisto example was novel drugs, and in this case we're talking about approved drugs. And there's a disease known as Niemann-Pick type C, which has been actually explored at the NCGC, and it's among many of the diseases for which Chris and his group are screening approved drugs. And this is just a slide to show you on the top, was all I really want to focus on, that there are many approved drugs for which screening could take place from the UK, from the US, and you can see there are quite a few that they have, but quite a few they still need to procure. So there are a lot of opportunities here for what we would call low-hanging fruit. One of the diseases for which low-hanging fruit might be relevant is chordoma, and chordoma is a disease that results from defects that occurred in the notochord during development. This is a rare disease that is not usually recognized early on, but slow-growing, locally aggressive tumors that often are observed later in adolescence or early in adulthood. There's a foundation that probably many of you are familiar with. Together with the foundation, expert groups have been assembled to try to help the drug discovery and development process, and have brought together patient cell lines to be screened. The main thing here to say is that 2,816 approved drugs have been screened with some promising hits. And follow-up studies are in progress with the goal of testing drugs in individuals with this disease from the approved drug collection sometime in the near future. Another thing I wanted to add here, though, is that if that doesn't work, this could also form the basis for novel drug discovery projects, because if there is a pattern seen by these approved drugs, for example, if all the drugs that are hits in these assays belong to one particular category, it may suggest that that pathway is something that should be explored and could form the basis for novel drug discovery. Okay, so now I'll just end with what was really trend. How can NIH help? We want to know how to help to translate the early findings from these promising tool compounds that might come from screening at NCGC or screening anywhere, or that come from finding approved drugs that have activity against potential targets in diseases for which there are no drugs. So one program then is this Therapeutics for Rare and Neglected Disease program that Steve and I are working on. And this was announced on May 20th, 2009 at NIH. And this announcement followed the approval of the budget and the history that Steve explained earlier in the talk. And this next slide just shows you the niche that Trend should fill. It's not clear that this is the only niche that Trend will fill, but right now what we're hoping is that we'll bridge, like the San Francisco Bay Bridge, the area from the NCGC through to the handoff to biotech or pharmaceutical, or to the clinical center to study. So Trend will be working in the probe to lead to candidate stage. We'll be studying these optimized chemical compounds in animals and in cell-based models so that we can develop drug-like molecules that could be the basis of investigational new drug applications, or INDs, that could be submitted to the FDA. And at that point, we hope, through having engaged these groups all along, we hope that biotech and pharma would be interested to take these forward after Trend and NIH had de-risked them early on. And we plan to work with biotech and pharmaceutical companies along the way so that whatever we're doing meets their standards, so that we don't come up with something at the end that isn't of quality that they would like to take forward. So we're pretty excited about this because it's even though pharma has been doing this for many, many years, the NIH hasn't done this in a big way for rare and neglected diseases. Many of the institutes have tried to do these kinds of things for specific diseases, but there's never been a trans-NIH initiative to do this for rare and neglected diseases. So I have here these slides that Steve looked at and talked through without them being in front of you earlier, so I'll skip through these. Just to say that this Trend initiative is a collaborative initiative governed by Steve and his group, but facilitated and administered at the NHGRI, and hopefully will be in close proximity to the NCGC. And we are in the planning stages of the staff and the facilities and the actual projects and the criteria for entrance of the projects. And also, we're in planning stages of how these projects will be assessed and how we would transfer them to big pharma or to biopharmaceutical companies. So I'll just end with something that is really relevant to this working very well, and that is something called the Grand Therapeutics Foundation, which I think is something that we're going to hear more about toward the end of the month at a meeting that's coming up. But there is a newly formed 501c3 called GTF, which will facilitate sustainable public and private enterprises for rare and neglected diseases. And this will be something that the details will be described in the future, but hopefully this kind of foundation will help individuals who have rare diseases or have an interest in rare diseases and want to facilitate drug discovery, will help those people get together and learn more about how to do this effectively and how to use resources that are common among these different groups to actually get drugs discovered and developed. So on the last slide, I just wanted to say we're trying to capitalize on new opportunities to make a difference to people who have rare and neglected diseases, and I think the time is really right for this, and we're really excited. We know the majority of diseases that affect humans are not being addressed, and we cannot continue using old models to do this effectively. And from a lot of viewpoints, including the discovery of the human genome and the subsequent insights that have been made, there's an unprecedented opportunity to develop new therapies to improve human health. And we're grateful that Zerhouni and Congress and others have put some faith in the NIH to do this as the New Therapeutics for Rare and Neglected Disease initiative. So that, I think, is all I really had to say, and I'm happy to take questions.
The first one, are there any possible risks associated with TR&D handing off drug candidates at an earlier stage? And if so, do you think that collaboration is required?
The risk of handing it off at earlier stages, I think that trends could potentially hand off compounds at any stage if someone were interested enough to take it forward. So that means that we could collaborate with entities at earlier stages before the IND if we felt comfortable that what we were doing was facilitating something and not losing it to follow up. And I think that the risk of doing that would be to lose it to follow up if the individual or the group that we handed it off to were really not interested enough to take it all the way. Because our real goal is to make sure that these go all the way to the clinic and then to approval if it's appropriate.
Okay, thank you. The next one is, how does NCGC choose which diseases to work on, and what is the process to explore projects with them?
So I think Steve mentioned this as well. This is a complicated question and a complicated answer because we are in the process of developing the flow of projects. So to not only develop what the criteria are for us to decide, but how projects will flow through our entities in order to actually be assessed. But the things that we will consider will be not just the disease, but the kind of compounds that are coming to us to be developed. So how well they look as early lead candidates, and how optimizable the compounds are. So those kinds of issues will be dealt with in the next few months so that we have very, very clear criteria for which to select not only compounds but the diseases that we're working on. Steve, do you want to add anything to that?
I think it's important to realize, too, that we will be having either a workshop or some other communication program that we can inform the public, and including the patient advocacy groups, the procedures and the processes that we will put into motion here to address this program and to initiate the program. So, of course, we would make the Genetic Alliance aware of this, and the communication will be going out from a number of sources. So when we're ready to go, I think you'll be getting a number of announcements about the possibility of either joining in live and in person, or joining in on a telecommunication of some type.
Okay, great. Our next question is regarding the efficiency or speeding of the development process. Is there a focus on FDA approval timelines, as in getting approval with less protracted phase 1, 2, or 3 approval process?
That's a great question. We would like to be able to do that, and we can't guarantee that, but all we can say is that in this process we're going to involve people with expertise in regulatory issues and the FDA as well, so that all parties are aware of the limitations of these small populations, and in the sense that not the usual path that I showed, the phase 1, phase 2, and phase 3, could be modified if need be in order to move things safely and effectively to approval. So we're thinking about that, but we don't have an answer, but at least we have people's attention.
If I could add too, during the past year, our office here, along with the Office of Orphan Products Development at the Food and Drug Administration and the review division, sponsored a seminar series. Actually, it was a continuing education series on the science of small clinical trials, and what we were focusing on were various statistical applications and study design issues that really needed to be discussed when you have small numbers of patients that you'd like to study. And then we're going to repeat this again in the upcoming year as well. But we had over 130 reviewers from the review divisions at FDA, and we had over 60 scientists and researchers from the NIH participate in this this 8-lecture course. And so we are trying to raise the awareness of the needs of the patient groups and the researchers who are investigating the rare disorders, and how to do studies a little bit more effectively with rare diseases and with the small populations. And so I think we're making some headways, and there have been also some very nice interactions with the European Medicines Evaluation Agency to work together to figure how we really can make this a much more efficient process of evaluating products for rare diseases. And I think if you look at FDA approvals recently, you'll see a lot more phase 4 type studies, and so people are suggesting that perhaps this is a way to go too, that we can collect so much information before the approval, but then the phase 4 after the approval, that we can continue to monitor the patients and the use of the drug and any potential problems that might arise, that we can look at this a little bit more closely and follow it. So, there are a lot of thoughts going into this, and so things are developing, and I think there's a lot more optimism now than several years ago, and so we hope to affect this within the regulatory and research communities.
Okay, and actually just pretty much answered another question without us having to ask it, but it was about whether or not research on diseases that are typically grouped, such as autoimmune diseases, would happen in that manner. So great. Okay, NCGC, about how many conditions are being addressed at the same time? Like how many through that program? Are there a number of conditions that are being studied at once right now?
So again, I'm not speaking for NCGC, but I know that they've got hundreds of collaborations, and they certainly are looking at multiple different diseases. But those projects are coming from the outside and are going through a separate type of review process.
Okay. What can be said at this time about the Grand Therapeutics Foundation and how that will be organized and populated?
I think I think that's a really exciting prospect, but again, that will be discussed later in the summer. There'll be an opportunity to understand more about that as these groups, all of these different groups, sort of coalesce a little more. So look for, I would say, stay tuned on that one.
Okay. And then,
but Steve, I think, had something.
Yeah, I think it's going to be discussed a little bit at the Genetic Alliance's annual meeting in July, I believe, and so there may be more information at that time on the July 17th meeting.
Okay. And what can the layperson do to help educate others and perhaps help with the venture philanthropy piece?
I can say from my perspective at the Cystic Fibrosis Foundation that the lay public has been extremely helpful because of the connection that all of the individuals in the community have with the people who have these diseases, and you can't underestimate the power of those interactions and those observations by people in the community about the disease itself or about potential therapeutics that they might think could be useful just based on knowing people with these diseases. So I think that it's always good for us to keep our ears open and to communicate in that regard, but also to help those individuals remember that educating potential donors and potential scientific contributors about how important it is that we do something about this, because there aren't pharmaceutical companies doing much about these things. So I think just increasing knowledge, increasing awareness, following our progress, following the progress of the entities that your disease is particularly focused in, just to keep being educated and give these groups some ideas of how to educate people well too.
Great. And then our next webinar coming up is July 7th, and that's part of our Strategies for Success. And what that is going to be about this time is educating members of Congress, and this ties in with our conference and our Hill Day, and you'll see that slide up there now. So our Genetics on the Hill Day is July 16th, and that webinar will kind of be preparing participants for that, but also even if you're not able to attend, it's kind of a great educational thing just to learn about educating Congress and some mechanisms to do that. So our conference is coming up July 17th through 19th, and our registration is still open for that. You can get to all the information about the conference through our website. And the day before the conference I mentioned is the Hill Day, and then the evening, in the evening just following the Hill Day, we have a really exciting night planned called Gene Screen, which is going to be a night of film on health and genetics. And so you can get more information about all that on our website. So thank you, everybody, and have a great day.
Thank you.
Thanks.